Hi everyone, my name is Adam Dede and I'm a postdoc in Beth Buffalo's lab at the University of Washington. This video is going to be the first video in what will hopefully be a long series of videos in which I'm going to try and summarize one academic journal article per week. The goal here is just to encourage me to read more and try and make some of this information a little bit more accessible. I'm not going to be trying to give any broad overview in any of these videos. Instead, I'm just going to be focusing on one article per week, kind of like a journal club. And it'd be great if anybody wants to give comments or feedback on any of their thoughts about these papers or about any papers that you'd like to see reviewed. So this week I'm going to talk about a paper from Wang and Barbas from last year entitled Specificity of Primate Amygdalar Pathways to Hippocampus. So in this study they used anatomical tracer injections into the amygdala, and the amygdala is highlighted here in this sagittal view of a macaque monkey brain. You can see that there's a cartoon of an injection needle, and basically what this is indicating is that they injected anatomical tracers into the amygdala, and then this arrow is showing you that their goal was to look at the connection from the amygdala to the hippocampus. And the hippocampus here is shown right behind the amygdala. Now, the amygdala is generally thought of as being important for emotional memories and kind of fear, arousal, that kind of thing. The hippocampus is generally thought to be important for the encoding of new memories and kind of declarative memory, the type of memory that you're aware of when you remember. So we all know from personal experience, if nothing else, that memories can have a lot of emotional content. And the question is, how does that happen? One idea is that the amygdala may influence the encoding of information in the hippocampus. So in this study, we're going to take a close look at what is the anatomy of amygdalar projections to the hippocampus. Over here on the right side, you can see highlighted on a sketch of the amygdala the 4 different injections in 4 different animals. And so this is kind of the starting point for these tracer injections that we're going to be looking at throughout this study. So the data that is going to be used throughout the rest of this presentation is all this type of thing that we see over here. So it's slices of hippocampal tissue viewed at high resolution under the microscope, either light microscopes or electron microscopes. And you can see in this blow up that there are areas that have little highlights on them. And these highlights in the hippocampal tissue are where those tracer injections diffused to when the amygdala was injected. And this gives us kind of a map of everywhere that is connected from the amygdala to the hippocampus. And you can see this quantified over on the right side, showing us that the anterior hippocampus tends to receive the vast bulk of the connections from the amygdala. And the posterior hippocampus obviously receives much fewer. So before we take a look at more specifically where these connections go to, let's just take a quick look at hippocampal anatomy. So here we're looking at a couple slices of hippocampal tissue taken in the anterior part of the hippocampus, which, as we just saw, is really the more important part when we're considering the amygdala projections. What you can see first of all is that the hippocampus is a really beautiful laminar structure. It has layers. And these layers have different purposes and different meaning if we see different patterns of connectivity in different layers. So you can see in the left side a Nissl stain that shows you where the cell bodies are, and on the right side an acetylcholine stain, which is showing you the density of a particular neurotransmitter. The dashed lines are showing us demarcations between different anatomical areas. So you can see that there's this kind of cornucopia-shaped thing, this circular thing, referred to as the cornu-humanus, and is split into the CA3, CA2, and CA1 regions. And we're going to be focusing on CA3 and CA1 throughout the rest of the paper here. And you can also see in the laminar structure, not only is there this cell layer that kind of wraps around, called the principal cell layer, or PCL, but there's also this area interior to that which is kind of devoid of cells. And that area is split into being called the stratum lacunosum moleculare, or SLM, and the radiatum, or RAD. And you can see in this cartoon area, this cartoon over here of neurons shown by these little triangles with lines coming off of them, that the dendrites to these neurons that live up in this principal cell layer come down into the stratum lacunosum moleculare. So it's really in that SLM layer that these neurons are receiving their input. So in both the CA3 and the CA1, the SLM is where the neurons receive their input, and then the PCL, or principal cell layer, is where the neurons actually live. So let's take a look now at where the amygdala actually projects to within these different layers. So here we're looking at points or axon boutons. So we saw in this slide that you can actually see the axon boutons, or endpoints of the axons, from the amygdala in the hippocampus when you view the tissue under the microscope. And here we're seeing little dots representing each axon bouton that was found when looking under the microscope in this particular slice. And you can see pretty clearly that the amygdala projections into the hippocampus are really very specific and limited to that SLM layer. So that is that input layer into the principal cells. So that's kind of cool that there's this kind of specificity to those inputs. So it suggests that the amygdala may have some role in modulating the input to the hippocampus. But let's see what more detail Wang and Barbas found. So not only does the amygdala project into this input layer, SLM layer of the hippocampus, but they also found at the electron microscopy level that the synapses from the amygdala are big. So here on the left we can see an example from electron microscopy where we see in black the axon terminal labeled with an AT here. And you can see in this particular axon terminal there's a mitochondria, which indicates a particularly large axon terminal. And then right next to it you can see a dendritic spine. So this is very clearly a synapse right here that we can actually see, which is super cool. And by looking at these synapses, they were able to actually measure what is the size of the axon boutons at these synapses. And what they found was that they tend to be right a little bit over 1 micron, both in the CA1 and the CA3 region when we're looking at these synapses that are coming from the amygdala. But in the same areas, they were able also to look at synapses that did not have a label. This black dye is that label, that tracer that was injected into the amygdala. And so they looked at other synapses that did not have that dye, and they can see in both asymmetric and symmetric different types of synapses that in the CA1 and CA3 the average size of a synapse is smaller than those that come from the amygdala. So not only are these amygdala inputs specific to the SLM input layer in the hippocampus, but they also happen to be larger than typical synapses. So let's see. Now, where are different inhibitory neurons in the hippocampus? So this is going to be a little bit of a detour to, again, we're going to review a little bit of anatomy. Although in this case, this is actually anatomical information that was not known until this paper by Wang and Barbas. So they just kind of took a side road and said, well, we want to know about inhibitory innervation from the amygdala to the hippocampus, and we don't really know that yet in the literature, so let's go ahead and do that, which normally would be a totally separate experiment, but they just threw it in here as an extra piece. So this is really a tour de force. And what they're showing us here is, again, we're looking at the hippocampus just really blown up. And they're showing us two different types of inhibitory neuron. So PV and CR, or parvalbumin and calretinin. And you can see here the CR in red and the PV in green. Now, just to think about the function of these two interneuron subclasses, the PV cells are able to enforce really strong inhibition because you can see that the PV cells live right in that principal cell layer on the outer edge of the hippocampus, and they're able to really clamp those principal cells down and stop them from being able to fire. The CR cells, by contrast, tend to live down in the SLM layer, and they also tend to have this effect where they actually inhibit other inhibitory neurons. So their net effect is to have specific ability to excite rather than to inhibit. And so it's kind of an inhibition of inhibition is a net excitation. So these CR and PV cells really have very different functions. And then the other thing that's kind of neat to look at here, you can see in this box labeled B and this box labeled A. So this box labeled B is up in the CA3 and the box labeled A is down in CA1. And you can see that in CA3 there's some green stain, so some PV stain, down in the SLM layer, whereas when we look over in CA1, the PV stain, that parvalbumin, is really limited only to the principal cell layer. So there may be some difference in how the amygdala is able to influence the hippocampus in these two different areas just by virtue of their different patterns of inhibitory neurotransmitters, or inhibitory neurons, sorry. So this is all great looking at the anatomy of inhibitory neurons, but do amygdala inputs actually synapse onto these inhibitory neurons? And in order to get at this, they went to the electron microscope again, and here again we're looking, you can see the axon terminals labeled in black, and now here these little black dots that you see inside the dendrites. These dots in the top two panels are stains for the CR calretinin. So this is an indication here that we are actually looking at an amygdala input in the hippocampus synapsing onto a CR inhibitory neuron in these top two panels. And then down here, the black stain in this dendrite is indicating the presence of parvalbumin. So this is a different stain in a different tissue sample. And so here we are actually looking at an axon terminal from the amygdala synapsing directly onto a dendrite on a parvalbumin neuron. So for sure, these synapses really do exist, which is pretty cool. And they went ahead and quantified it at the level of the electron microscope. And as you might have expected from the different pattern of CR and PV interneurons in the CA3 versus the CA1, we can see that in the CA3, so that was this area up here, in the CA3 there's this mix of both CR and PV interneurons receiving innervation from the amygdala, whereas in the CA1 it's only the CR neurons that are receiving innervation from the amygdala and not the PV neurons at all. Now, one thing to note is that on this y-axis these percentages are quite small. Now, this is because most of the innervation from the amygdala to the hippocampus is excitatory. These inhibitory interactions between the two structures are a small percentage of the total innervation, but likely play an important role. So now, just to kind of put it all together a little bit, if you think about how the amygdala is innervating the hippocampus in this bottom panel, this is a figure that you've seen before, you can see the SLM layer is receiving most of the input. And then up here you can see that it's this SLM layer up in the CA3 in this panel labeled B that has both PV and CR interneurons being labeled, whereas down in A it's really only the CR neurons that are labeled in the SLM layer. And this then fits with the fact that the CA3 is receiving input onto both PV and CR interneurons, whereas the CA1 is receiving input only onto CR. So how might this actually come together in order to influence the processing of information in these two areas? So in CA3, if we imagine amygdala input coming in, and there are going to be some principal cells that receive direct input from the amygdala and some principal cells that receive no direct input from the amygdala. Now, these ones that receive direct input from the amygdala, they're going to get excited by this input from the amygdala, but then also these CR interneurons and these PV interneurons will also become excited by the input from the amygdala. Now, these PV interneurons, they're going to just clamp down all of the excitation in the area, so they're inhibiting everything. The CR interneurons are going to be, in a very targeted way, inhibiting inhibitory interneurons. So in this way, they're going to affect a kind of release from inhibition. So what's going to end up happening is, as a net effect, the amygdala will now be able to tune the output of CA3 because it's going to be exciting these particular cells that it has direct excitatory input onto, but then it's also going to be causing inhibition broadly, which will stop other neurons from being able to fire, and in this way it'll be able to tune the output. By contrast, in the CA1, since it doesn't have this broad inhibitory effect that we see caused by the PV innervation, it's only going to be able to bias output onto a particular set of principal cells, but it won't have this ability to also simultaneously inhibit other principal cells. So it won't have quite the same tuning ability. Instead, it'll be more kind of a biasing towards a particular type of output, or at least this is one way that these results might be interpreted. So that's it, and I hope you guys enjoyed the video and learned something. So thanks, and hopefully I'll see you next week.